Students, we are going to start a new chapter today, that is cell cycle and cell division. So, before starting the chapter, we should know that what is the content of this chapter, ki ase. We have to talk about cell cycle in details. The next point is the cell division boli kole ami jigita types of cell division janu. Basically, we talk about two types of cell division, that is mitosis and the meiosis, mitosis in details, meiosis in details, and also to some extent a little bit about mitosis. Mitosis basic details na lage because it is almost like we have already discussed binary fission. So, mitosis not in details, but mitosis, meiosis in details. So, basically we have to study only about three things, that is the cell cycle, the mitosis process and meiosis process, right. Now, to begin with, the word called as cell cycle, right. Cell cycle mane ki? What is cell cycle? Ki boli koba? Exactly, all the events, right. All the events that occurs in a cell, at a cell we know what is what does a cell do? A cell will obviously after getting matured it will divide, no. So, cell eta before dividing tar preparation ki, divide keneke kore, how does a mother cell splits into two cell, right. All these events are totally included in something that is called as a cell cycle, and obviously cell cycle duta part of divide kora ase. One is called as the interphase and the other step would be called, or the other part would be called as the dividing phase, right. So, cell cycle ki hoy, nothing but the events, right. Hey, events kini kot hoye ase, everything is occurring in a cell. The cell will prepare itself first and only then it will go for division. As far as division is concerned, it can be either mitosis or it can be meiosis. Basically, when we talk about cell cycle, we are concerned with mitosis, karon jano mitosis e basic kya hoy, meiosis to it's only confined to some cells, right. It is also called as reductional division. So, ami first ki korim, cell cycle or definition to likhim and then we will go. So, first likha, what is cell cycle? Cell cycle or likha, an orderly sequence of events by which a mother cell duplicates or replicates, eketai bostu, duplicates or replicates, aru ki koribo duplicate? Its cytoplasmic content and genetic material and after duplication ki hobo, and passes them faithfully into two daughter cells. So, proper definition boli kole, this is what is the definition of a cell cycle. So, what is cell cycle? It is nothing but some series of events, right. Aru ei events bilak kot hoy ase, the cell that is going to divide and the cell that is being that is going to divide, obviously we can use the word called as a mother cell. So, mother cell e divide kora homoy ji bostu bor hobo, no, obviously cytoplasmic contents bilak baribo, genetic material has to get doubled and then it will go to the daughter cell. So, this is the proper definition. The next point about cell cycle is, it is a genetically controlled method. So, cell cycle ki hoy, genetically controlled method. Third point is, it varies from organisms to organisms, right. So, what we understood by the third line? So, what is cell cycle? We understood is nothing but some certain types of events that occur in the mother cell. This cell cycle is controlled by the genes. Next, so organisms or we know living organisms are made up of cells, but cell cycle process too, depending on that organism, it varies. As far as duration is concerned, no, kisumanor besi somoy lage to complete that cycle, kisumanor kom somoy lage. And when we are talking about variation, we have to remember certain examples, like in case of human beings, the cell cycle is of 24 hours. Tinta example monot rakhibo lage. In case of yeast, it is 90 minutes, and in case of bacteria, it is 20 minutes. So by these examples, what we understood is je cell cycle to ki hoy, vary hoy, right? Car cell cycle by which cell is dividing, depending on that, the duration would be different. So in other words, what we can say, in case of human beings, eta cell or duta cell hobo lagile, what is the duration? It is 24 hours. Similarly, in case of yeast, if the mother cell has to divide and form two daughter cells, what is the time period? 90, and similarly in case of bacteria, it is 20. It's very less in case of bacteria, right? Ekhiloke kiba doubts? Okay. So remember the examples properly, okay? So here questions ahe, the specific time duration to upor question ahe. So we need to remember the time duration. And maximum question has been asked about yeast and bacteria. Okay. Now we will be talking about phases of cell division or phases of cell cycle, right? Can I rub the bone? Okay. 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 Now we have to talk about the phases. By the word phase, what we understand? Okay, before writing about it, let's talk about cell cycle in a diagrammatic form. All of this I'm bhalke diagram to dim. So if you are talking about a cycle, this is the cell cycle. So a cycle to ki thakibo, kisuman phase thakibo, no? So we have certain parts, and out of this, one part, right, is called as, I think you will remember, monot ase niki ki asile? M phase, G1 phase, S or G2, the entire cell cycle has been divided into 4 phases, right? Phases ba 4ta step thake. Tar bitorot this 3 phase, G1, S and G2, tak ami ki boli kom? Interphase. And this part is called as the dividing phase, but division phase. Starting, swap. By the word division phase, starting kuntu cell or hobo, if you are talking about that cell, no, kune divide koribo, mother cell divide koribo, right? So mother cell, so A2 part hole tumar division phase. Division of yate ki thakibo janne? Mitosis or ami jibilak porisilu, no, prophase, metaphase, anaphase, telophase, right? So if you are talking about a cell here, yate eta cell ase. A cell to divide kori belai ki form koribo, duta cell form koribo. So this cell will undergo all this process, and ultimately the daughter cell will enter in the G1 phase. From G1 it will go to S, from S it will go to G2, and finally the cell is again ready to divide. So ami gradually bostu bilak likhilo, right? But what you remember is je once this mother cell is ready ketia hobo, eta cell divide koribole ready ketia hoy, jetia tar prophase, metaphase, anaphase, telophase, all these phases are completed, right? So when all these are completed, the daughter cell will progress in the same method. Okay, now talking about that diagram, no, jiman bor 4ta phase dekhilu, no, all of us rakhi dim. আমি যিমান কেইটা টার্মস দেখিলো কি দেখিলো M phase দেখিলো G1 দেখিলো S আৰু G2 right so we have seen 4 phases but all those 4 phases has been broadly divided into only 2 phases if the question is asked the cell cycle or phase কেটা cell cycle or phase দুটা one part or one phase is called as interphase and the other phase is called as the division phase or the dividing phase so when we talk about phases of cell cycle কেটা থাকিব chart format likhi lua we have 2 phases one is called as I phase or interphase so eta part of I phase or interphase boli koy aru eta part of ki koy M phase or division phase so the cell cycle is divided into 2 phases the I phase and the M phase right kisman points right basically I phase or M phase what is the difference or what we understand M phase as the name suggests we are talking about that actual cell division right either we are talking about mitosis or we are talking about meiosis so mitosis or jimanbur steps thakibo ba jimanbur events hoy no jenga prophase metaphase anaphase telophase and similarly in case of meiosis also meiosis 1 and meiosis 2 all those things are included in the M phase then what is included in the I phase eta cell division koribo lagile if it has to undergo mitosis it has to prepare itself right it should be ready and only then the cell will be able to divide or form 2 daughter cells hey ready je hoise divide kora agote nijoke nije ready koribo libo it you can compare it as like this na by taking an example suppose you want to go somewhere before going out or before attending a marriage or you are going for a party you have to make yourself ready right that phase is called as interphase so what happens in interphase the cell becomes ready for division what happens in the M phase the cell undergoes division is the difference understood right okay S phase I phase of G1 G2 and S phase M phase of ki hobo se M phase of tumar actual process of division to hobo eneke bujha na je party eta le goi asa cell eta divide kora agot jikhini phase hoy hetu interphase koy interphase nuhua loke cell to keneke divide koribo eta bostu kiba eta bostu hobo lagile tar kane preparation lagibo no that preparation phase is called as interphase prepare hole the cell will say now I am ready to divide jetialeke hetu kotha nokoy no je I am ready to divide tetialeke division no and whenever we are talking about division we are talking either about mitosis or meiosis so mitosis meiosis war agote cell to capable hobo lagibo no mitosis hobo lagile capable in the sense means what if we are talking about a cell that mother cell will form 2 daughter cells hey daughter cells or mother cell or jikhini contents asile that contents needs to be what evenly distributed to the 2 daughter cells so enough material thakibo lagibo hey cellot so these steps are nothing but the preparation phase means what the cell should be capable to divide and only after that it will divide jeneke party eta tumi ready hobo lagibo tar pisot he tumi jaba ready nuhua ke tumi jabo nwara to buji paisa okay so one is preparation phase and this is the actual thing or the cell division phase next if I ask you out of these 2 phases right both the phases are important obviously preparation is also important and division is also important actual cell cycle by actual cell division boli kole which phase we are talking about cell division boli kole we are talking about this phase right actual division to kot hoy actual division to M phase ot hoy preparation to kot hoy preparation to I phase ot hoy that's the first point second point regarding these 2 phases is as far as the time period is concerned as we have discussed no ami ki koisilu the depending on organism to organism or depending from cell to cell tahator cell cycle or duration bilak different hoy right. For example, in case of human beings, we have seen that it takes what 24 hours for one cell to divide and form two daughter cells, right. So, we have discussed those examples. So, eta cell cycle or 24 hours lage or maybe 20 minutes lage or maybe 90 minutes lage, but both the process should occur within that specific period itself. So, in case of human beings, if the cell cycle is of 24 hours, within that 24 hours, I phase o hobo lagibo, M phase o hobo lagibo. Is the concept clear? Buji paisa? Okay. Now, we have not done in details, but even after that, at a question jodi hudu, as far as these two phases are concerned, kuntu phase or you think is longer or will take more time? Kuntu phase basic homoy lagibo, division phase or ne I phase or basic homoy lagibo. It's always the I phase, yeah, it's always the I phase that it will take more time as compared to the main phase. Means what? Simple kotha no, ami prepare hobole bahut homoy lao, 3 dinor pora agor pora bhabo, okay, we have a party on that day, so we need to dress ki, this ki, that ki, jam keneke, ki korim. So, all this, it takes a longer time. Biakhanot gola, goi pele khai pele gusi aile, it's a very shorter time, right. So, out of this, it's the I phase that is longer as compared to the M phase. So, we are going to write certain points. I phase, likha, it's also called as into data hoise roba. The first point about I phase is, likha, it is the resting phase. Board clear hoise ne? Also called, yeah, also called preparatory phase. It could be also be called as the non-dividing phase. Next, this phase is considered as metabolically most active phase of cell cycle. And 95% of the cell cycle is of I phase. Right? Similarly, in case of M phase, this phase is said to be the most dramatic phase. Karon actual drama kon kot hoy, division or drama kon yat hoy. Next point, it takes only 5% of the cell cycle. Gutei cell cycle to jodi ami 100% boli consider koru, tetia hole 95% kune occupy koribo I phase and 5% would be of the actual phase, or actual phase boli noko, the dividing phase. The third point is, this phase has 2 steps. And what are the 2 steps? Ami janu already, division of the nucleus and the division of the cytoplasm. The words that we use is karyokinesis aru cytokinesis. Just go through the points. So, talking about the cell cycle. So, we are talking about this cell cycle. This cell cycle is divided into 2 phases. One is called the M phase and the other is called as the I phase, right? I phase basic homoy lage, M phase kom homoy lage, if you are talking about in terms of cell cycle. What happens in the I phase? The cell prepares itself for division, and so it is called as the preparatory stage. It is also called as resting phase. And why is it called as resting phase? Because division does not start in this phase, right? Non-dividing phase, because by division we understand mitosis or meiosis, where thus mitosis and meiosis occur, it will occur in the M phase. Out of the cycle, 95% of the cell cycle is I phase and it is only 5% that belongs to M phase. So, ইয়াতে কিবা doubts আছিল নেকি? As far as this 95% and 5% is concerned, আমি এটা example দি পেলাই will make it more clear. Once you are done, please say so that I can rub the board. This part is over, I suppose. Okay. Hetu partu hoise niki ne nai hua? ঠিক আছে হ'ব লিখা হৈ গ'ল okay so 95% আৰু 5% টো অকমান just point বুলি লিখি পেলাই লিখি লোৱা we take an example of the human cell. In humans আমি কি পঢ়িছিলো cell cycle or duration কি আছিল যে এটা mother cell to divide into two daughter cell, what is the time duration? 24 hours, right? And this 24 hours, suppose we are considering it as 100%. Then I phase kiman hobo 95%, right? Which is approximately equal to 22.8 hours. 22.8 hours could also be considered as K, 23 hours. So, out of the 24 hours, I phase or kiman? 22.8 hours. And if you are talking about M phase, it is 5%, which is approximately equal to 1 hour. Right. So, as we have discussed, what we said is cell cycle or Ruta phase, one is called as the M phase and the other is called as the I phase. Talking about both the phases, longer phase kuntu, it is always a preparatory phase that is supposed to be longer, right. 95% is I phase and 5% in case of M phase. Yata a question ahibo pare, in case of a human cell, I phase by M phase কিমান hours কারণ আমি জানো এটা human cell if that cell has to divide to form two daughter cells, the maximum requirement is 24 hours. Out of this 24 hours, 23 almost 23 hours it takes for the preparation phase, and within 1 hour it completes the division. Is it understood? So ekhini saba okay so I phase 23 almost and M phase is just 1 hour. So, this is all about the phases. Now, details koribo laibo phase bilakor. Next likha we have to write something about I phase according to Howard and Pelly, what they said, interphase has or is divided into 3 phases, that is the G1 phase, the S phase and the G2 phase. Right. Now you can draw the diagram. We are talking about it as a cell cycle. Titya hole G1 phase, S phase, G2 phase, a gutekhini ki boli kom M phase that will consist of ki prophase, metaphase, anaphase or telophase, and we have another marking ekhini okuman dotted area ke disu. This is called as G0 phase, right? From G by jot telophase he hoise no he line dalor pura goi jot prophase arambho hoise, this part is called as interphase. Is the diagram clear? Broadly divided in two phases, the dividing phase and the interphase. Interphase of K bhagot bhag koriso 3 types, that is, or 3 phases, G1, S and G2. But in the diagram, in the G1 phase, we have seen another part that is called as G0 phase, right? It is a question to ahe, the interphase of K-ta phase, then you cannot be saying it as it has 4 phases, G0, G1, S and M. The answer to that question would be interphase comprised of 3 steps, that is G1, S and G2. G0 to ami tetia kobo noaru. G0 phase is a part of G1 itself. Is the diagram clear? Mot likhisu prophase, metaphase, anaphase, telophase. Ami generally mitosis to consider kora hoy. Thik ase? So jumbilak mitosis or phase bilag no prothom step ki asile prophase, tapsot metaphase, tapsot anaphase, or lastly telophase. Telophase ot ki hoisile je duta cell ultimately form kore no. Prophase, metaphase, anaphase, telophase, the entire portion is M phase or the dividing phase. Nai porwa etia porham he prothomote to ami G0 is a part of G1 itself, right? So he karone moi dotted area ke mark korisu karon basically divide korise ki 3 ta phase of G1, S and G2. G1 ore eta part of ami G0 hisabe mark korisu, and what is the significance of G0 will be discussing. Khali yate jonto interphase likhisu, just take care that it starts from where end of telophase, telophase or jonta line akisa tar pura goi pelai beginning of prophase no e gutei section kinik ami ki boli kom interphase boli kom. Okay, now from examination point of view, what is important is first you need to see the diagram. Diagram based question ahibo A, B, C, D, D dibo, and it's a very easy question, so we'll understand the which phase it has been marked. Next is in details we have to talk about each and every phase. Talking about M phase, obviously mitosis in details and meiosis in details, the entire process we need to talk about. But talking about interphase also, G1 details, S details or G2 details. What we know is, so a gutti interphase actually hoy ki the cell prepares itself for division. So when the cell prepares itself for division, within that preparation period, no, bahut changes occur koribo cell tut. So you have to remember what are the specific changes that occur in each phase. Out of many changes, no, specific ketaman change ase, that's very important. Whenever you talk about G1, you have to remember some stages. Whenever you talk about S, again some points to be remembered. When you talk about G2, again certain points to be remembered. Question keneke ahe je S phase ot ki hoy bahut bostu hoy tar bhitor eta sobotse important bostu hoy. If you have read or if you have gone through the chapter, if you know something called as DNA duplication. So DNA duplication eta cell divide kori pelai duta cell hobo, duta cell hua loge loge when the cell was I mean if suppose we are talking about this cell, right, this cell will behave as a mother cell, this cell will divide and form 2 daughter cells, right. A daughter cells will have the contents of the mother cell itself, right. So yare jikini cytoplasm ase, yate jikini genetic material ase, tare to eta part ahibo, right. So it has to increase its contents, or increase its contents kot hobo in the interphase. যেতিয়ালৈকে ইনক্ৰিজ নহয় তেতিয়ালৈকে to divide হ'ব নোৱাৰে no because it will not be able to divide it properly. So it has to, this mother cell needs to increase all the contents and after completion of the increase nucleus genetic material বেছি হ'ব লাগিব ইয়াত যিমানবোৰ cytoplasm আছে organelles আছে বাঢ়ি যাব লাগিব তেতিয়াহে after that কি হ'ব division হ'ব. So the next part that we are going to study is what happens in these three phases, mitosis, meiosis, later. As of now we are talking about interphase. So ekhini jedi hol heading koriba G1 phase. So what happens in the G1 phase? Can I rub the diagram? Ekinloke, is it clear? So what we have learned, cell cycle is nothing but certain types of events that occurs in the cell, right, before division and or after division also. Next, the entire cell cycle divided into two parts, that is the interphase and the dividing phase. Interphase is longer. It consists of three parts, that is G1, S and G2. Firstly, G1 phase. So what happens in G1 phase? Some points about G1 phase. G1 phase is also called as first gap phase. G is for gap. Next, so G1 phase মানে কোনটো phase কথা কৈ আছো diagram to সদায় মনত ৰাখিবা হা we are talking about this M, this is G1, this is S and this is G2. Okay, so this phase, first gap phase. Next, the second point is a phase to আমি কি বুলি কম, post mitotic phase বুলি কম ne pre mitotic phase বুলি কম? Are you understanding the question? Yeah, obviously. So it will be also called as what? Post mitotic, because mitosis will yate ki hobo telophase hobo, so mitosis will get over and after mitosis what happens is G1 phase and so it is called as post mitotic phase. Next, it is also called as pre-synthetic phase. Kwa? Yeah. Oh. Okay, doesn't matter. Kwa, please ask. Exactly, daughter cells too, they will enter the G1 phase. Karan hey daughter swap, this is the mother cell, it has formed two daughter cells, this daughter cell will again divide. So to divide again, akko cell cycle to jabo lagibo na lage. Understood? So what we understand is if you are talking about division, no, this mother cell suppose ekhini jagat asile, a mother cell to change hoy ultimately a to stage oloke ki form korise, duta cell, এই দুটা সেল কোড এন্টার কৰিব G1 ত, so it is called as post mitotic phase. Obviously, exactly, এই দুটা সেলে আকৌ তাক নিজকে প্ৰিপেয়াৰ কৰিব, তাৰ পিছতহে এই দুটা সেলে আকৌ দুটা সেল ফৰ্ম কৰিব. Understood? Okay, it is also called as pre-synthetic phase karon S phase to kami synthetic phase boli kon pisot kom. Next, one important point about this is maximum cell growth occurs here. মই যেনেকৈ কৈছোঁ ন প্ৰত্যেকটো ফেজৰ কিছুমান পইণ্টছ আমি as far as writing is concerned we'll be writing so many points, right, but from examination point of view কেইটামান point বেছি important. So ইয়াৰ পৰা question আহে যে if you are talking about all these phases কোনটো ফেজত cell to maximum grow কৰে, the maximum cell growth occurs always in the G phase. So this is a statement that is very important. We know that if the mother cell wants have to divide into two daughter cell, it has to grow. By the word grow, what we understand, the contents bilak bahibo lagibo, no? Contents bahibo lagibo, size or dangor hobo lagibo. So growth sobotse besi kot hobo G1 phase ot. Next point, no duplication of genetic material. If a cell has to divide into two daughter cells, then its cytoplasmic content should also increase, and together with that the genetic content should also increase. Jiman cytoplasm ase tatke beshi hobo lagibo, organelles bilak beshi hobo lagibo, aru tar uporiu nucleus or genetic material the amount should also increase. But this increase in the genetic material does not occur in G1 phase, it occurs in the S phase. So, the question that is asked is duplication of DNA is related with which phase of cell cycle? It is related with S phase of cell cycle. Another point, another way of putting the question is the S phase or so what's the important step to key? S phase of key eta bostu hoy jotu ki very important for the cell cycle. S phase of DNA duplication hoy. So, yate question ketiba etu ahibo pore, does DNA duplication occur in G1 phase? No, DNA duplication does not occur in G1 phase, means genetic material to increase no hoy. Is the point understood? Okay. Next, then what happens in this phase? Synthesis of amino acids, proteins, RNA, nucleotides, baki burji bostu ase. Energy rich compounds, etc. Ebilak bostu tat ki hoy? Synthesis hoy. And by the word energy rich compounds, we are talking about what? ATP, GTP, etc. Ebilak sob G1 phase of hoy. ইয়াতো synthesis হয় সাবলে কলে প্ৰত্যেকটো ষ্টেজতে synthesis হয় ওকে G1 ৰো কিবা কিবি synthesis হ'ব S2 হ'ব আৰু G2 টো হ'ব কিন্তু that word no S phase S is something that's denoted with the word called as synthetic phase and that is মানে কি out of all this phase যেহেতু genetic material S phase of সবতছে বেছিকে environment কি অকল S phase ওতে increase কৰে so we by the word synthesis we are concerned with that synthesis of the genetic material কিন্তু as far as the other compounds are concerned এটো G1 টো হ'ব G2 টো হ'ব আহিব সেল অৰ্গানেলিজ বিলাক আহিব তাৰ পিছত আকৌ ATP আহিব সব আহিব right next one RNA polymerase and enzymes for DNA synthesis becomes active in this phase. And the last point is it takes maximum time of all the phases. So, when we talk about G1, egita point monot rakhibo laibo. So, G1 is called the gap phase. What is the most important point of G1? That maximum cell growth occurs in this phase. The cell has to grow and only then it will become ready to divide. In this phase, certain substances like amino acids, proteins, nucleotides, ATP, GTP, etc. Eblakoru synthesis hoy. RNA polymerase and enzymes that are responsible for DNA synthesis. Karon yate dekhisan na yate genetic material duplicate hoy nai no. So next phase ot S or sobotse important point ki je yate DNA duplicate hobo. DNA duplicate koribo lagile ba DNA amount to badhibo lagile obviously certain types of enzymes would be responsible. Hey enzymes bilak active code koribo in the G1. After it becomes active it will go to S and in this phase the contents will divide. So, I have to doubts ase niki. As far as RNA polymerase is concerned, second year course of when you will go for genetic classes, genetics of eblak details of, right? You will be talking about something called as transcription. Jot niki ki hoy? Transcription porhisa jano nai porha nohoy? এটা ডিএনএৰ পৰা আৰএনএ বনোৱা প্ৰচেছটোক আমি কি বুলি কওঁ ট্ৰান্সক্ৰিপশ্যন বুলি কওঁ ৰাইট এইবিলাক আমি জেনেটিক ক্লাছত পঢ়িম right so RNA polymerase the main enzyme no that is responsible for the process called as transcription etu code active hoy G1 phase of active hoy No, your voice is not clear, sorry. I did not hear. Last point too. It takes maximum time of all the phases. Of all the phases, we understand G1, F and G2. Right? It interpreter maximum time code lage G1 phase of lage. Oh, sorry. Spelling no. PHGE কলে এখন ভাইরাস হয়ে যাব ন জানো ন ফেজ PHGE ফেজ পার্টিকল বায়োলজিক্যাল ক্লাসিফিকেশন কৰিছো আমি okay it takes maximum time of all the phases so these were the points about G1 next লিখিবা S phase S phase is called as synthetic phase. So what's the important event ki hoy? DNA duplication occurs here. Bracket eta bostu likhi thoi diason DNA duplication occurs here very very important point but the chromosome number okay do you have any idea regarding chromosome genes then DNA etc etc genetic material boli kole what you understand eta cellor genetic material kot thake nucleus right nucleus ki thakibo chromosomes right chromosomes chromatids these are the things that we need to study in the cell chapter right ultimately hey chromatids bilakot ki thakibo genes DNA etc talking about a chromosome from the cell chapter diagram educator diagram on ase nai something like this second point too okay DNA duplication occurs here then put a bracket DNA duplication occurs here but the chromosome number remains same the হেতু বুজাব লাগিব ৰবা কিন্তু বুজোৱা গৈয়ে নাই এতিয়ালৈকে নহয় ক্ৰোমোজোমছ বিলাকতহে ডিএনএ থাকে ক্ৰোমোজোমছ বিলাকতহে জিন থাকে okay so what we understand so this is again from the cell chapter remember a diagram like this chromosome boli kole this is the most common diagram that we remember right it has two parts or two arms that are called as chromatids or a chromatids bur eta pointer log lagi thake that is called as the centromere I think you remember all this right so what we understand by the duplication of DNA so etu eta chromosome this is also a chromosome right yate ki thakibo DNA thakibo yate ki thakibo DNA thakibo yato ei dutat ki thakibo DNA thakibo ei dutat ki thakibo DNA ekdom simple way to remember or to understand the statement okay so what we said DNA duplication occurs here we know that cell to jetia divide koribo tar genetic materials khini bahibo lagibo right so genetic materials jetia bahe we are talking about nothing but DNA duplication when we talk about DNA so this is a chromosome this chromosome will have the genes ot ki thakibo DNA bur thakibo so ebilakot jetia genes thakibo genes bilakot DNA thakibo what I said what happens in the S phase the DNA content increases ta mane agote ekhini asil aru ekhini asil etia ki hol double hoi gol le nai yate DNA ki asil ekhini aru ekhini yate dudal hol a very simple way to remember or to understand so ultimately what we have seen the content of DNA has doubled but if you talk about chromosome etu ki hoy edal chromosome etu ko ki boli kom ami one chromosome etu dudal chromosome ne edal chromosome so eta centromere ase centromerot chromatids bilak lagi ase this diagram represents chromosomes now the point is how many chromosomes eta ne duta chromosome it is just one chromosome if I draw like this another diagram then this one is one chromosome this is another chromosome ditya hai ami a pair of chromosomes boli kom is the point understood common diagram a2 jetia DNA duplicate kore then we have a diagram like this right no doubt the DNA has duplicated but the number of chromosomes remain the same so this is the meaning of this statement in the S phase ki hoy DNA duplication occurs but edal chromosome or pra dudal hoi na jay chromosome edale thakibo kintu chromosome dal thicken hoi jabo prothomote thin asile etia thick hobo thick kyo karone hoise karon tat DNA contents kini beshi hoi goise is the concept clear everyone so jeti ami chromosome aku no chromosome diagram to ami actually detail cell of sam ha ako ami jetia hei korim right actually ami cell to this is a portion that we should cover it in cell so chromosome boli kole this is also a chromosome a2 chromosome aru a2 chromosome right etu keita chromosome one chromosome yato keita chromosome one chromosome jedi aru eta diagram aku something like this etia hale etia ami ki kom two chromosome a step to clear hoise nai chromosome eta it is denoted by a diagram like this where it has two arms aru a arms bilakok ki boli koy chromatids boli koy hebilak pisot details of korim right so chromosome diagram kenekwa enekwa chromosome ki thake the two chromatids are joined at a point and that point is called as a centromere so what is the diagram of a chromosome this one or it could be this one also what is the similarity between these two diagram 1 and diagram 2 the similarity is this is also one chromosome this is also one chromosome what is the difference between the two diagram yate DNA content kom ke ase yate DNA content beshi ke ase the second point is this chromosome you can also say this chromosome is thin this chromosome is comparatively thicker why is it thicker because the contents of DNA has doubled yate jiman bur DNA ase right yate beshi thakibo na thake right so chromosomes are a2 this is also a single chromosome this is also a single chromosome but if you are talking about this diagram it talks about ki a pair of chromosomes is the point clear so S phase or ki hoy chromosome number increase kore ne DNA content increase kore it is always the DNA content that increases and not the chromosome number is the point clear eta chromosomot two chromatids a pair of chromatids but during cell cycle ami porhim pisot cell division ot ketia enikwa stage ahi jay thik ase jetia DNA duplicate koribo no tetia hale amar ebilak ami beshi ke dekha pam chromatids bilak karon DNA beshi hobo laibo no yate beshi hoi na jay ami jetia mitosis details ot kori jam ako ebilak discuss korim right as of now এই বস্তুটো ক্লিয়ার হইছে S phase ot ki hoy chromosome number beshi hoy ne DNA content beshi hoy beshi is not the word that we use the word called as we use a word that is called as doubling of DNA or doubling of chromosomes. It's obviously doubling of the DNA content, right? Our DNA content or ki hoy? Generally it is, when we talk about DNA content, piece of details kori mami. When we talk about DNA content, it is generally denoted by 2C. 2C. A2 DNA content kiman? 2C. Jeta double hoy jabo, A2 DNA content kiman hobo? 4C. So question to ahibo pore 2C pora 4C kuntu stage ot hoy? 2C pora 4C DNA S phase ot hoy. Is the point clear? Yes or no? Everyone? So, etia basic complex ke bhaibo na lage. What you need to understand is in the S phase, the most important event is DNA duplication occurs here. Right? Next, yate S phase or aru kisuman important bostu hoy. For example, Centrioles duplicate. Centriole word to shunisa nai agote? Cell porisa nai? Cell porisa? Right? So, enuka eta diagram dekhisa, if you are talking about this one as a nucleus, nucleus er usorote eta horu ke eneke dot eta thake na thake? A rough diagram. Aru etu kami ki boli kau? Centriole. Aru etu hol nuclear membrane er usorot thake, ha? Or etu kami ki boli ko nucleus. So this is the structure that we are talking about, the centriole. First point, what is a centriole? It is nothing but a microtubule. Ebila words janibole hole you have to go back to the cell chapter, right? Centrioles, key important point, if you are talking about the basic difference between the plant cell and animal cell, centriole is present only in case of animal cell. Cells, right? Next, what is the function of a centriole in a cell? Yate why? Yate ami function, it helps in cell division, yes, but to be proper, how does it help in cell division? Okay, kya 7 or porisa cell porha nai niki? Cell chapter to porha nai niki? The cell to obviously centriole ase. Okay, so first point ami ki gom palu, centrioles duplicate and centrioles are present in animal cell. From cell cycle point of view, what is a centriole? What is the function of a centriole? These are not relevant with this chapter, right? A duta question or answer ami kot pam, cellot pam. But in this chapter, what is important? Question ahe, in which phase of cell cycle does the centriole duplicate? It duplicates in S phase, right? Aru centriole ki hoy, but what is the function of a centriole? The main function of a centriole is it helps in the formation of the spindle rays. But spindle formation not help kore. Spindle formation mane cell division of spindle formation hoy na no hoy? Something like this. Etu diagram dekhisan nai? Suppose ami metaphase or kotha koi asu. Ei gute structure to ami ki boli kom? Spindle, right? So spindle formation hoy, spindle formation or pisot ki hoy, chromosomes bilak separate hoy jay, right? But chromatids bilak separate hoy jay aru duta opposite polole gusi jay. Ei spindle fibers bilak form koribole kune help koribo? Centrioles. Kuntu point or pra develop koribo? Centriole or pra develop koribo. Eta cellot spindle fiber, if you are talking about in this diagram, what is the number of centrioles that you see? 2. This is a centriole. This is a centriole. At the very beginning, when we are talking about a single cell, eta cellot, before division, centriole keita thake? Eta, at the starting phase. Gradually, the centrioles doubles. Duta hobo laibo, karon duitar pora ki hobo? Spindle fiber bro originate koribo. A doubling of the centriole occurs in the S phase. Is the point clear? So, centrioles duplicate. Next, chromosome, in S phase, chromosome consists of 2 chromatids, but the duplicate chromosome does not become distinct. So, this phase is also called as invisible phase. So, অকমান এইখিনি বুজিবলে হলে সেলৰ অকমান information you have to remember. What we are saying here is starting from a cell, if you are talking about a cell, so etu ki hoy nucleus. Nucleus of ভিতৰত কি থাকে genetic material থাকে, genetic material in the form of কি chromosomes, chromatids, whatever you say, right? If you remember prophase, অকমান prophase ত কি হয় প্ৰথমতে nucleus to কেনেকে থাকে ball of wool, right? Woolor ball etar nisake thake na thake ekku bostu distinct nohoy, all the threads are interwoven amongst themselves, nothing is clear kuntu chromosome code ki hoise chromosome bilak distinct hoy janu prophase ot just try to remember. Okay, as of now what you remember, eta cell jitia cell division or prior to cell division, the nucleus or the genetic material that is inside the nucleus remains in a interwoven form. Interwoven form mane nothing but you can compare it as a ball of wool. Wool or ball eta jeneke round hoy thake no, strands bilak ki hoy thake interwoven hoy thake, edal separate ke thake niki wool dekhisan nai wool bilak sweater eta banale jinbilak ball ahe no, wools ball bilak he wool bilak ki hoy etar major eta round ball hoy thake, right? You see it in a ball form, you do not see it separate, right? Separation chromosomes bilako ki hoy, chromosomes ba chromatids ba genetic material jiye kwa prior to division genetic materials bilak ki hoy in the form of a ball thake, nothing is distinct clearly, right? A chromosomes ba genetic material bilak gradually they will thicken and ultimately you can see them in the form of threads that is called as chromosomes. Prothomote eku dhoribo nwara, lahe lahe gradually ta distinct hoy jay ar tetia ami chromosome to identify koribo paro. Before preparation chromosome to me identify koribo nwara, right? So what happens in S phase? S phase is a part of what? Interphase. Interphase or ki hoy ase, the cell is preparing itself to divide, right? So nuclear material duplicate korise jedio you cannot see the 2 chromatids of a chromosome, mane proper ke tumi ei diagram to dekha nopua. DNA duplicates, so jeri DNA duplicates ei diagram to ami nucleus or bhitor dekhibo lagisile kintu proper ke ami dekha na pao, and so this phase is called as invisible phase. Is the point clear? Just think, in the S phase what happens is DNA duplicates, right? DNA duplicates means what? From 2C it reaches 4C. If DNA duplicates, the diagram of the chromosome will also change. Duplicate koribo lagile tat chromatids basically thakibo lagibo, right? Enukwa eta diagram hobo lagisile after duplication of DNA, but this is a phase that we do not see or it does not becomes distinct in the S phase, and so this phase is called as invisible phase. Chromosome likhibo para, genetic material likhibo para. And the last point about S phase is, in this phase histone protein gets synthesized. So ako question ahe, histone protein ক'ত synthesize হয় histone protein S phase. Talking about histone protein, again details about histone protein in the genetics class. As of now, just in one simple sentence, if you remember, if you want to know what is the function of a histone protein, no, at a protein, hey protein to naam ki histone. What is the function of the histone protein? It helps in packaging of the DNA. Packaging of the DNA means we know DNA is a long thread-like structure, right? Eta cellot bahut thread থাকে and the length of this thread is very very long. So iman dighol bostu eta iman horu cell etat keneke thakibo, right? So it has to be kunto histone or packaging of DNA. Just hey duta word likhile hol karon later details ot poriboi lagibo. ঠিক আছে কেনেকে প্যাকেজ কৰে কেনেকে প্যাকেজিং কৰে হেবিলাক বস্তু ডিটেইলস ত পঢ়িম আমি জেনেটিক্স ক্লাছত as of now what is the function of the histone protein packaging of the DNA means what we understand by the word packaging of a DNA is a very long strand right বহুত দীঘল ৰছী এডালৰ নিচিনা এই ইমান দীঘল ৰছীডাল এই সেল অকমানি সেল এটাৰ নিউক্লিয়াছৰ ভিতৰত কেনেকে থাকিব তাক ফোল্ড কৰি ভালকে প্যাকেজিং কৰি পেলে তাক মাই থৈ দিয়া and who helps in packaging is a specific protein that is called as histone protein code synthesis হয় it gets synthesized in the S phase. So these are the points about S phase. So up till now what we have studied is interphase consists of 3 phases, that is the G1 phase, the S phase and the and the G2 phase, right? One mother cell after it forms 2 daughter cells, the daughter cells will also divide again. In order to divide, the daughter cells needs to cross over these phases G1, S and G2. Tetiahe daughter cell akko divide koribo paribo. What is important is what are the events that occurs in G1? What are the events that occurs in S and G2? As of now, G1 or what is the most important point? The maximum cell growth occurs in this phase. Aru G1 or ki hoy? Je kisuman dhoroner proteins, kisuman dhoroner amino acids, nucleotides, ebilak bostu tat form kore. So here duta hol important point about G1. Talking about S phase is supposed to be very important because DNA duplication occurs here. Nucleus or junkhini genetic material thakibo that needs to get doubled so that it could be distributed equally between 2 daughter cells. So DNA duplication occurs in the S phase. Another important point is centrioles that is present in the animal cell that helps in spindle fiber formation and astral ray formation. That structure gets duplicated in the S phase. Next, histone protein also gets synthesized in the S phase. Our invisible phase, just word to mone rakhiba ketiba hudibo pare je kuntu phase ok invisible phase buli koy. It is the S phase that is called as invisible phase. Any doubts? Okay. Next likha G2 phase. Again certain types of events that occurs into G2 phase. It is called as the second gap phase. First gap phase ki asile G1. What happens in this phase is cytoplasmic organelles such as mitochondria, chloroplast and Golgi bodies বা Golgi complex ji likha are doubled. Next, in this phase, synthesis of RNA and proteins continues. Continues word to kyo likhiso? Karon we have seen this function in the G1 phase also. Next, spindle protein, and when we talk about spindle protein, we are talking about tubulin. A word to mone rakhiba. Synthesis occurs in this phase. Next, ester formation also occurs in this phase. Ete ester boli kole ki bujo? Okay, we'll explain. First, let's write. Next point likha, the cell prepares itself to enter into the M phase. Next point, synthesis of some protein, synthesis of protein, samne likh le hobo, protein kinase and synthesis of protein kinase for regulation of cell cycle. Point kita likhi lo, then we'll explain. For regulation of cell cycle or cell division, cell division would be proper. Synthesis of protein kinase. Kinase bula at a specific protein A2 phase of synthesize hoy. And what is the role of kinase? It helps in regulation of the cell division process. Aru kotha patim, we will be talking about kinase and CDKs. So other points clear, board con clear hoise ne? G2 phase is called as second gap phase, not very important from examination point of view. Cytoplasmic organelle protein, kinase is a protein. ठीक है एक protein टोर नाम की kinase. So question खुदिबो परे kinase protein code synthesized होय. It gets synthesized in G2 phase. ठीक है organelles बिलाक double होय याते, right? Synthesis of RNA and protein continues. Why continues? Because these things also occur in the G1 phase. Important point about G2 phase is tubulin protein code synthesis hoy. Tubulin protein G2 phase of synthesis hoy. What is the function of this tubulin protein? So A2 phase at a specific protein called as tubulin, or at a specific protein called as kinase. Ebla protein or tat ki hoy synthesis hoy. Tubulin protein or function ki it helps in spindle formation. Hey protein to thakile he spindle fibers bilak form koribo, right? So A protein to code form kore in G2 phase, right? Ester formation also occurs here. Now talking about ester, by the word ester, what we understand, a star shaped structure is called as ester, right? So cell etat nucleus thakibo, nucleus or usort ki thakibo, centrioles thakibo. Centriole pura we have seen, say, নাই হোৱা exactly because contains to same cell or ভিতৰতে organelles বিলাক duplicate কৰা আৰম্ভ interphase তে হৈ যায় ঠিক আছে so cell division বুলি যেতি আমি obviously enlarge হ'ব cell division বুলি কোৱা আগতে এটা cell divide কৰা আগতে it has to get enlarged কাৰণ তাৰ contents বিলাক তো বাঢ়ি গৈছে right so এটা diagram যদি আমি এনেকৈ আঁকো ন এটো ধৰা mother cell এটো mother cell এ দুটা daughter cell form কৰিছে ঠিক আছে এটো এটা diagram আৰু এটা diagram এটো mother cell ই ডাঙৰ হৈছে ডাঙৰ হৈ পেলাই দুটা ডটাৰ সেল ফৰ্ম কৰিছে এটা কোনটো ডায়াগ্ৰাম ইজ কাৰেক্ট ফ্ৰম সেল ডিভিজন পইণ্ট অফ ভিউ a mother cell forming two a mother cell forming two daughter cell a mother cell forming two daughter cells right but what is the correct diagram obviously this one because we understand the mother cell needs to get enlarged it has to increase its contents and only then it will go for division right so jehetu sob bostu bari jabo tetla obviously cell to enlarge hoy jabo clear clear no okay now I was talking about ester formation so ester formation ki hoy kisman star or nisna no star like structure is seen and this is what is called as ester so I think you remember we have talked about the two centrioles bilak already s phase or duplicate ho goise right a centrioles bilakor pora ki hobo spindle fibers bor ilabo right jibilak ni chromosomes ot log lagi thakibo right from this centriole no kisuman star like structures enukwa dhornor microtubules ba kisuman enukwa dhornor tube like structures ulai that looks like or that takes the appearance of a star and this is what is called as ester right so ester formation ester ki hoy eta star like structure code form kore centriole form kore kihe form kore what is responsible for forming these lines no it is nothing but microtubules and microtubules from the cell chapter ako eblak relation code ase cellot so we need to read the cell chapter first and then we have to come to the cell division but since you all asked me to like discuss cell cycle first and then we'll go to cell he karone okman kheli mili hoise ami jodi properly ahilu heten যদি সেল আগতে পঢ়িলো হয় তাৰ পিছত সেল সাইকেল পঢ়িলো হয় তেতিয়াহলে এবলা কনফিউজন নহলে হেতেন ৰাইট ওকে এনিৱে সো centrioles ot ki thake in and around eta star like structure thake a star like structure to ami ki boli ko ester so ester formation code hoy ester formation G2 phase ot hoy right any doubts up till here okay from cell chapter at least once you have studied the cell chapter right do you remember something called as microtubules microfilaments intermediate fibers etc hunisa niki words bilak all these are related with what all these are related with the cytoskeleton of a cell eta cell it has a skeleton right because of that skeleton it has a proper shape and size right that cytoskeleton who forms the cytoskeleton microtubules microfilaments and intermediate fibers so microtubules ki hoy kisuman tube like structure enuka bostu microfilament kisuman thread like structures right so ebilak bostu e ki form kore eta structure ba skeleton form kore cytoskeleton oh exactly hebilakor cytoskeleton thakibo তোমালোকে এটা বস্তু পঢ়িছিলা সেলত যদি মনত আছে কার্ট হুইল লাইক স্ট্রাকচার পঢ়িছিলানে লাইক দিস বেসেল বডি আৰু সেন্ট্ৰিওলৰ যোনটো স্ট্রাকচার হেবিলাক কিহেৰে বনোৱা আছে হেবিলাক মাইক্ৰোটিউবলছৰে বনোৱা আছে এস্টাৰ কোনে ফৰ্ম কৰিছে মাইক্ৰোটিউবলছে ফৰ্ম কৰিছে এস্টাৰ কোড ফৰ্ম কৰিছে নিয়াৰ দ্য সেন্ট্ৰিওল অৰ সেন্ট্ৰিওলত কিছুমান ইলংগেটেড থাকিব আৰু কিছুমান শৰ্ট থাকিব এই শৰ্টখিনিক আমি কি বুলি কৈছো ester clear okay coming back to this portion important is tubulin protein importance is kinase baki khini to okay clear yes please ask present in centrioles kole bhul hobo karon centriole is nothing but a ki boli kom it's a microtubular eta pair of centriole boli koy ei pair to ami centriole boli kon juntu niti eta organelle mazot thake that is called as centriosome eta organelle তাক আমি সেন্ট্ৰিওসোম কওঁ সেন্ট্ৰিওসোমত এনেকুৱা দুটা মাইক্ৰোটিউবুল থাকে এই দুটাক আমি কি বুলি কওঁ সেন্ট্ৰিওল বুলি কওঁ সেন্ট্ৰিওলছ বিলাকৰ পৰা কি হয় সো এনেকে না কি পেলাই যদি এনেকে আমি সেন্ট্ৰিওল দুটা we just depict it in the form of a diagram from the centrioles ki hoy spindle fibers bilak ulai right a spindle fibers bilak ki hobo they will remain attached with some of the spindle fibers will remain attached with the chromosome and some will remain just like that right ekhini ami amane so ha eta centriole opra ki ulabo kisuman fibers ulabo right eta cellot keta centriole thake দুটা we have already talked about it right cell নাই পঢ়াৰ কাৰণে প্ৰবলেম হৈছে সো এটো হল centriole বা কি ওলাইছে কিছুমান ফাইবাৰস ওলাইছে এই ফাইবাৰস বিলাক আমি কি বুলি কওঁ spindle fibers বুলি কওঁ if you are talking about this spindle fibers if you see the diagram properly some spindle fibers are attached with the chromosomes and some spindle fibers are not attached with the chromosome ডায়াগ্রাম তো ভালকে চোৱাচোন এডাল ফাইবাৰৰ লগত এটো ক্ৰোমোজোম লগ লাগি আছে কিন্তু এডাল ফাইবাৰৰ লগত ক্ৰোমোজোম আছে নেকি এটোটো নাই সো এটা সেন্ট্ৰিওলৰ পৰা যিবিলাক স্পিন্ডল ফাইবাৰস ওলাই ন এবিলাকত কি হয় it has been divided into two types one is called as continuous fiber and the other is called as discontinuous fiber continuous fiber কোনটো কম এটা সেন্ট্ৰিওলৰ পৰা আনটো সেন্ট্ৰিওলকে যোনটো continuously spindle fiber that stretched হৈছে without touching any chromosome that is called as continuous fiber discontinuous kuntu the spindle fiber that remains attached with the chromosome so what we have learned centriole thake duta tar pra spindle fibers bi ulabo and in and around the centriole you also see a star like arrangement this arrangement is made up of certain types of microtubules or a star arrangement to kami ki boli ko ester a ester formation ketia hoy jetia spindle fiber formation hoy during cell division is a point here everyone okay so this is all about the three phases of interphase but as I said no তিনটা ফেজ আছে কিন্তু তার লগতে আৰু এটা ফেজ আছে that is called as G0 phase if you remember that first diagram that we have drawn এটা dotted area আঁকিছিলো no so if you are talking about this diagram this is the M phase this was G1 S and G2 ইয়াতে এটা dotted area আঁকিছিলো this is called as a G0 phase right so we need to talk about the G0 phase now what is so important about the G0 phase what happens is in this case no can you see the diagram properly okay so yate ki asile mother cell after completion of this division phase became two daughter cells these two daughter cells will enter G1 phase right এতিয়ালৈকে বাৰু G0 নাই ঠিক আছে দুইটা ডটাৰ সেল কোড এন্টাৰ কৰিব G1 phase so that they can divide and form two more cells but what happens is out of the daughter cells that enter in the G1 phase each and every daughter cell may not divide again means what mother cell forming two daughter cell this daughter cell may divide this daughter cell might not divide is the concept clear eta mother cell e duta daughter cell form koribo duita daughter cells e divide koriboi lagibo not necessary right some of the daughter cells they escape the cell cycle they don't want to divide so if they don't want to divide they will require a place to stay kot thakibo tahat karon yar pora to already ulai ahile দুটা daughter cell form korile duta daughter cell form korile it is bound that they will have to come to G1 but after coming to G1 some of the daughter cells felt that they won't divide if they won't divide then obviously they will not go through this pathway যদি সেলটোৱে divide কৰে তেতে G1 ৰ পিছত S S ৰ পিছত G2 G2 ৰ পিছত M লৈ যাব যদি যিকোনো এটা ডটাৰ সেলে ডিভাইড কৰিব মন নাই তেতিয়া হলে হিটো এই সাইকেলটো কমপ্লিট নকৰিব তেতিয়া what happens is it comes to G1 and then it goes back to another phase that is called as G0. So G0 phase is a part of G1 phase. Who stays in the G0 phase? Those cells that do not want to divide or those cells that want to escape the cell cycle. The meaning is the same, right? As far as these cells are concerned, so duta yale ahile, eta cell divide korile, eta cell yale gusi gol. Juntu cell G0 phase ot gusi gol, he cell tue may not divide or will divide whenever the necessity comes. Is the point clear? So likha nai moi etialoke prothomete bostutu buji lua tar pisot ami likhim. So G0 phase ki eta enukwa phase jot niki cells bilak accumulate hoi thake kenuka dhoroner cells accumulate hoi thake jun bilak cell cell cycle ot na jai ba jun bilak cell cell divide nokore. Not necessary each and every daughter cell will divide. The cell that does not want to divide will always remain in the G0 stage. Is the point clear? Everyone? Okay. Okay. First point. Ki kolu? Yes, please ask. হেতুৱে কৈ আছো ফাৰ্স্ট টু G0 ত কোন বিলাক সেল থাকে বুজি পালো যোন বিলাক সেলে ডিভাইড কৰিব মন নাই হে বিলাক সেল G0 ত থাকে simple way to understand. Next point, the cells that are present in the G0 stage remain in this phase temporarily or permanently. যোন বিলাক সেলস ইয়াতে আছে হে বিলাক সেলসে ডিভাইড নকৰে ডিভাইড একেবাৰে নকৰে নে কেতিয়াবা কৰিব কিছুমান সেলস এনেকুৱা যোনে নেকি ডিভাইড কেতিয়াও নকৰে permanently G0 phase ত থাকিব আৰু তাৰে ডিভাইড কেতিয়াও নকৰে কিছুমান সেলস এনেকুৱা আছে যে এতিয়া মোৰ মন নাই ডিভাইড কৰিবলৈ মই পিছত ডিভাইড কৰিম যেতিয়া নেসেসিটি আহিব বস্তুটো বুজি পাইছা নে নাই So if suppose this is G0, right? G0 stage ot thora ketaman cell ase, right? A cells bilak ki hoy? They have escaped cell cycle. Means what? They will not divide, right? Out of these cells, there are certain cells that will never divide. Tahate permanently thaki jabo. Right? And out of and the rest cells they will divide এই কনসেপ্ট বুজি পাইছা নাই সোৱা G0 phase এটা phase তাৰ ভিতৰত কেনেকুৱা সেলস বিলাক থাকিব কেনেকুৱা ডটাৰ সেলস থাকিব এনেকুৱা ধৰণৰ ডটাৰ সেলস বিলাক থাকিব যোনে নেকি ডিভাইড কৰিবলৈ মন নাই এতিয়া মন নাই ঠিক আছে একেবাৰে মন নাই নে এতিয়া মন নাই যদি কেতিয়াও ডিভাইড কৰিব মন নাই তেনেকুৱা সেলস বিলাক these cells will never divide এইবিলাক cells they are at this moment in the G0 but they will divide whenever there is a necessity এই স্টেটমেন্টটা ক্লিয়াৰ হৈছে নে নাই escape cell cycle বুলি কোৱা হৈছে কাৰণ এই দুটা ডটাৰ সেল যেতিয়া G1 ত আহিব G1 ৰ পিছত S ত যাব S ৰ পিছত G2 যাব সো তাহাতে সেল সাইকেলত সোমাই গল কিন্তু ইয়াতে যোনবিলাক সেল আছে তাহাতে যেহেতু এটো সাইকেলত যোৱা নাই so we have written the word called as escaped the cell cycle means they will not divide clear okay so as we have discussed in the G0 stage the cells will remain there either temporarily or permanently if the cells remain permanently then they will never divide so kisuman cells tate permanently thakibo ei permanently thaga cells bilake ketiao lifetime divide nokore only they will grow kisuman cells temporarily thakibo junbilak cells divide koribo when the necessity comes as far as examples are concerned okay What you have seen is, take a very simple example. Talk about the hair. Amar hair ba suli hore ne nohore? Suli bila hore ne nohore? Hore? Hori ami ki topa hoi jao niki? Normal cases, except certain types of exceptions, that is different. Normally amar to daily cell hore, suli bila hore ne nohore? কিন্তু চুলি সৰা পিছতো আমি টপা হৈছো নেকি? আজি আমাৰ জন্মৰ পৰা ইমান বছৰ হ'লহি তোমালোক টপা হৈছা নেকি বা আমি টপা হৈছো নেকি? নাই হোৱা। কিয় নাই হোৱা? Obviously growth হৈ আছে, right? সেই চুলিখিনি যেতিয়া সৰিব তাৰ প্লেছত আকৌ সেলটোৱে ডিভাইড কৰিব, right? সেই জেগাতে কৈ আহিব লাগিব যে চুলি সৰিলে বা স্কিন শেডিং, কিছুমানৰ স্কিন শেডিং হয় ন, ড্ৰাই কণ্ডিশ্যন বা ঠাণ্ডাৰ দিনত কি হয়, ছালবিলাক কি হয়, ড্ৰাই হৈ পেলাই ফ্লেক্সৰ নিচিনাকৈ ওলাই আহে, ৰাইট? সেইখিনি সময়ত কি হ'ব, আকৌ নতুন ছাল ডেভেলপ কৰে। সেই নতুন ছাল বা নতুন চুলি ডেভেলপ কৰিব লাগিলে কি হ'ব, সেলটোৱে ডিভাইড কৰিব লাগিব নালাগে, ৰাইট? চ' জিনত ষ্টেজত এনেকুৱা ধৰণৰ চেলছ কিছুমান থাকে যোনবিলাকে ডিভাইড কৰিব, কিন্তু যেতিয়া তাৰ নেচেচিটি হয় তেতিয়াহে ডিভাইড কৰিব। চুলিখিনি সৰিব লাগিব তেতিয়াহে চুলি ফৰ্ম কৰিব। কোনে ফৰ্ম কৰিব? ইয়াত থকা চেলছবিলাকে ফৰ্ম কৰিব। স্কিনটোৰ অকণমান শুকান হৈ পেলাই ওলাই যাব লাগিব তেতিয়াহে নতুন স্কিন ফৰ্ম কৰিব। কোনে ফৰ্ম কৰিব? নতুন স্কিন ফৰ্ম কৰা মানে নতুন স্কিন চেলছ ফৰ্ম কৰা নহয় জানো? ইটছ দ্য চেম থিং, right? So who will form those? The cells that are present in the genome. So in other way around, what you can say, do you remember a word called as quiescent center? আমি rootsot পঢ়িছিলো anatomy. Quiescent center, do you remember? এনেকুৱা এটা ডায়াগ্ৰাম আঁকিছিলো, right? আৰু ইয়াতে মাজতে quiescent center. Quiescent center মনত আছে নাই? এটা এনেকুৱা group of cells where the cells remain there or accumulates there up till the time they are required to divide. যেতিয়ালৈকে তাহাঁতৰ ডিভিজনৰ দৰকাৰ নাই তাহাঁতে তাতে accumulate হৈ থাকিব. That is what is called as quiescent cell. Quiescent center ক'ত দেখিছিলোঁ? root anatomy দেখিছিলোঁ. In the root tip or in the center তাতে এটা inactive cells কিছুমান থাকে, right? এই চেলছবিলাক কেতিয়া এক্টিভ হ'ব? যেতিয়া দৰকাৰ হ'ব। Exactly the same in this case. G0 phase is also called as quiescent phase. Why? Because here the cells will remain and they might divide whenever the necessity arises. But there are certain cells that will never divide again. Is the point clear? Okay. এতিয়া এটা কাম কৰোঁ, আমি G0ৰ কিছুমান points লিখি লওঁ, maybe that way it will become more clearer. So লিখা আছে G0 phase. G0 phase ot likha, it is also called as resting or dormant or inactive phase. So remember this word. Yeah. চেলছবিলাকে ডিভাইডহে নকৰে কিন্তু গ্ৰ' নকৰিব নেকি? চেলছবিলাকতো লিভিং হয়, লিভিং চেলেতো সদায় গ্ৰ' কৰিব, ৰাইট? ডাবল ডাবল থাকিব এণ্ড মেজৰ অৰ্গেনছবিলাকত সেইটো থাকিব। ফৰ এক্সাম্পল, ইফ ইউ আৰ টকিং এবাউট আ হাৰ্ট চেল, হাৰ্টৰ আমাৰ যোনবিলাক চেলছ আছে সেই চেলছবিলাক কেতিয়াও ডিভাইড নকৰে, কাৰণ উই নো হাৰ্টটো আমাৰ কি হ'ব, হাৰ্টটো আৰুতো ডাঙৰ হৈ চেলটো ডিভাইড কৰি 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 ডাঙৰ হৈ হয়তো যাব নোৱাৰে, হয় নহয়? ঠিক তেনেকৈ ইন কেছ অফ নিউৰনছ, নাৰ্ভ চেল, সেইবিলাকে ডিভাইড নকৰে। যিমান নাৰ্ভ চেল তোমাৰ জন্মতে আছে এতিয়াও সিমানেই নাৰ্ভ চেল আছে, হাৰ্টত যিমান চেল বেবীত আছিলে এতিয়াও সিমানেই আছে। As far as the number of cells in the heart is concerned, এটা বেবীৰ হাৰ্টত যিমান চেল আছে আমাৰো হাৰ্টত সিমানেই চেল আছে, but the size of the baby heart and the adult heart varies because the cell grows but do not divide. Is the point clear? No. Daughter cells ট্ৰান্সফাৰ কৰিব দৰকাৰেই নাই ন যেহেতু ডিভাইডেই নকৰে তে ডটাৰ সেল সব বস্তুতে যে ডটাৰ সেল ফৰ্ম কৰিব লাগিব ইচ এণ্ড এভৰি সেলে যে ডিভাইড কৰিব লাগিব নট নেচেসাৰী যদি প্ৰত্যেকটো সেলে ডিভাইড কৰিলে হেতেন তেতিয়াহলে আমাৰ দেখোন কি হল অৰ্গানছ বিলাক জাস্ট থিংক এবাউট দ্য অৰ্গানছ that are present inside a body অৰ্গানছ বিলাক দেখোন আজি যদি if the heart comprise of 10 cells কালি 20 cells পৰহিলা 40 cells বাঢ়ি বাঢ়ি দেখোন এটা লাম্প অফ হাৰ্ট হৈ যাব সো কিছুমান এনেকুৱা জেগা আছে য'ত নেকি সেলছ বিলাক নিজে নিজে ফৰ্ম হ'ব কন্টেন্টছ বেছিকে থাকিব আৰু সেই অৰ্গানছ বিলাক ফৰ্ম কৰিব কিন্তু ডিভাইড নকৰে সেলছ বিলাক প্ৰত্যেকটো সেলৰে গ্ৰোথ হ'ব কাৰণ প্ৰত্যেকটো সেল ইজ এন এক্সাম্পল অফ আ লিভিং সেল okay this is a mother cell দুটা সেল ফৰ্ম কৰিলে ঠিক আছে এই সেলটোৱে দুটা সেল ফৰ্ম কৰিলে এই সেলটোৱে দুটা সেল ফৰ্ম কৰিলে সাপোজ এই সেলটো ডিভাইড কৰা নাই ঠিক আছে এটো হল ই দুটা ফৰ্ম কৰিলে এটো এই সেলটো যোনটো নেকি ডিভাইড কৰা নাই হি সদায় এটো শেপতে থাকিব নে এনলাৰ্জও কৰিব সেল গ্ৰো কৰিব তো সেল গ্ৰো কিয় কৰিব কাৰণ তাৰটো অলৰেডি তাৰ কন্টেন্টছ বিলাক ডাবল হৈ আছে ন ডিভিজন বুলি ক'ব নোৱাৰা হি দুটা সেললে স্প্লিট নকৰে তাৰ ভিতৰতে যিখিনি কন্টেন্টছ বেছিকে আছে হি তেনেকেই থাকি যাব বুজি পাইছা এইটো ষ্টেজলে আহিবলে লওঁতে এই সেলটোৱে যেতে এইটো সেল ফৰ্ম কৰিলে তাৰটো কন্টেন্টছ অলৰেডি বেছিকে আছে সাইটোপ্লাজমিক কন্টেন্টছ বেছিকে আছে ডিএনএ কন্টেন্ট বেছিকে আছে ৰাইট তাৰ বা অৰ্গানেলিজ ডুপ্লিকেট অলৰেডি হৈ গ'ল right kintu a cell to divide nokore in the sense it will not get splitted into two cells a two cell jeniki it got splitted it shared it contains a cell to share nokore hi tar nijor bhitorte dangor ke accumulate kori hi thakibo okol size of grow hoy bas হেকাৰণে মই যোনটো ক'লো ন এটা হাৰ্ট দুটা হাৰ্ট যদি কম্পেয়াৰ কৰা ন বেবিৰ হাৰ্ট আৰু এডাল্টৰ হাৰ্ট তাত যদি নাম্বাৰ অফ সেলছ চোৱা তেতিয়াহ'লে বেবিৰ কিমান সেলছ থাকিব এডাল্ট হাৰ্টতো কিমানেই সেলছ থাকিব কিন্তু বেবিৰ হাৰ্টটো সৰু এডাল্ট হাৰ্টটো ডাঙৰ এই ডাঙৰটো কেনেকৈ হ'ল গ্ৰ' হৈছে কাৰণে ডিভিজন হৈ পেলাই ডাঙৰ হোৱা নাই দ্য নাম্বাৰ অফ সেলছ ডিড নট ইনক্ৰিজ দ্য সাইজ ইনক্ৰিজড Okay, coming back to this, G0 phase of keep on rakhiba inactive phase. This phase is considered as inactive phase. Remember this word. Next point. Here, the cells escape cell cycle. A pointed already buzai dilu. Next, cells will not divide or redivide but they will attain maturity divide nokore kintu tahat matured hobo mane tahate function kori thakibo next point cells in this phase remain here either temporarily or permanently. Examples of the cells that remains temporarily are skin cells. Another example could be hair cells. Jetia dorkar hoy tahate divide kore. Permanently junblak cells divide nokore. Please ask. Or redivide. Sorry. Okay. Or redivide. The cells that are present in this phase will not divide or re-divide, but they will attain maturity. Tahat matured hobo, কিন্তু divide nokore. The cells in this phase may remain temporarily or they will remain permanently. Examples of certain cells that always remains undivided, heart cell or neuron. Aru kisuman cells june niki necessity hole divide koribo from the G0 phase, it is temporarily, it is skin cells and hair cells. G1 phase to agot ahibo. Eta cell, mother cell jeta duta daughter cell form koribo, duita daughter cell G1 phase ote ahi jabo. Kintu G1 phase ot ahar pisot they will decide they want to divide or they don't want to divide. If they want to divide, from G1 they will go to S. If they don't want to divide, they will stay in one corner of G1, that is called as G0. Teneke to aru ghor nai, no, they don't have compartments. So it is within the G1 stage where certain cells do not divide. They do not go to the S phase. Hetuke ami ki buli koiso, G0 phase buli koiso. Clear? Okay. Next lekha, cells from, continue, no? Cells from G0 phase goes to G1 phase. Cells from G0 phase goes to G1 phase whenever required. G0 phase to jete heading korisila no, yate aru eta word likhasun quiescent. It is also called as quiescent phase. Quiescent. Right? So daughter cells bilak kole palehi, G1 ot palehi. G1 ot pai pelai, if they want to divide, they will go to S and then to G2 and then to the division phase. If they do not divide, they will stay in a phase that is called as, that phase is called as G0 phase. Now, JD, all the daughter cells do not want to divide, tete hale ki hobo? Means my question is, uncontrolled G0 cells will cause what? Forms ki? Jedi cells bilak, daughter cells bilak G1 phase or pra goi pelai G0 ot accumulate kore, tetia hale ki koribo je tahate ultimately accumulate kori what they will form is, they will form a tumor, right? Obviously, it will be a dangerous situation. Hekarne cell division koribol. Uncontrolled G0 cells. Jedi G0 cells ki hoy? Jiman thakibo lage kintu besi hoy jay. Prottektu enukwa eta hoy gol. The situation where the daughter cells refuse to divide. Tetale ki hobo? Eta chaos form koribo no? Ultimately, that accumulation of cells will ultimately form ki tumors. So ultimately, body to ki develop koribo tumor. Oh, genetic hobo. It can be hormonal, it can be genetic, it can be anything. Kiho karne tenukwa phase hoise tar root cause to ami janibo lagibo. Right? So I think that's all for today. Right? So today in the class what we have discussed is, we have talked about the word called as cell cycle. Cell cycle ki hoy? Nothing but certain events that occurs in a cell, right? Divided into two parts, that is the interphase and the dividing phase. Which one is longer? Obviously the interphase is longer. Next, if you are talking about the duration of the cell cycle, it varies from organism to organism. We have given 3 examples. Talking about interphase, it consists of 3 phases, that is G1, S and G0 phase, right? Tarpisot, out of these phases, certain major events that we need to remember from all these phases, and what are the events we have discussed, right? Sometimes what happens, once the daughter cells reach the G1 phase, it might not divide. If it divides, it will go to S. If it does not divide, it will remain in a phase that is called as the G0 phase. In the G0 phase, the cells will remain either temporarily or permanently. Permanently means they will never divide again. Such examples are heart cells and nerve cells. And as far as temporary is concerned, the cells will remain there up till a certain time. তাহাতে ডিভাইড এটা সময়লৈকে নকৰে, কিন্তু কেইদিনমানৰ পিছত তাহাতে ডিভাইড কৰিব। কেতিয়া কৰিব? যেতিয়া নেচেছিটি আহিব। Right? So by this, I think with this, I'll wind up my class today. We'll continue it in the next class. Thank you.